All right, it's 2:30. I'm going to get started. If you are just filing in, it's okay. I have a little bit of a introduction to get through, so feel free to still get yourself settled. I want to welcome everyone to the Great Lakes Phragmites Collaborative. And if you're counting, we're on our 53rd webinar since we've started this webinar program. I'm Teresa, and I'm joined also by my colleague Sam Tank, and we're hosting today's webinar on behalf of the Great Lakes Phragmites Collaborative. And our presenter today is Nic Tippery from University of Wisconsin-Whitewater, and they will be presenting using molecular genomic tools to investigate genetic diversity and hybridization in Phragmites. So I'm very excited to hear the talk, and I'm sure you are too. Just as people start filing in, I'm going to give a little bit of a housekeeping items to review. For those who have came to our webinar talks before, it should look familiar, but for anyone joining us today who's not familiar, I want to introduce the Great Lakes Phragmites Collaborative. This collaborative, which we acronym to the GLPC, is a partnership to link people, information, and action on invasive Phragmites. And the collaborative is coordinated jointly by the Great Lakes Commission and the USGS Great Lakes Science Center. And the collaborative facilitates communication, among stakeholders across the region and assists in the development of management tools and programs, and serves as a resource center for information on Phragmites biology, management, and research. And the collaborative is led by the Multi-Agency Advisor Committee and applies the principles of collective impact to advance our common agenda for Phragmites in the Great Lakes region. And here's what our common agenda looks like. And the Great Lakes Phragmites Collaborative has been working towards advancing these member strategies that are outlined here, which you can see on the screen. And the common agenda is one of the big steps we took towards aligning our regional goals and establishing a shared vision for change. And the purpose of our common agenda is to clearly define the intentions of the collaborative, develop a shared understanding of gaps and problem areas surrounding Phragmites, and highlight the work in areas and strategies we want to work on or wish to work on in the future. And if you have any questions of what the Great Lakes Phragmites Collaborative does and want to be involved in some of our work, you can learn more by visiting our website. And it's one of the big components of our work, and one of the ways we engage with Phragmites managers, is through the Phragmites Adaptive Management Framework, or as we call it, PAMF. And PAMF utilizes participatory science to provide data-driven Phragmites management guidance through an adaptive management framework. Basically, what this means is we just want to establish a way to learn from what Phragmites managers are actually doing and turn that data into annual guidance to help managers make effective and efficient management decisions. So if you want to learn more about the PAMF program, you can visit our website, which is there on the screen. Also, you can get in contact with the PAMF coordinator by emailing them at pamf@glc.org, and they'd be happy to answer your questions about what that program looks like if you're interested. And if you or anyone in your agency would be interested in getting Phragmites-specific management guidance, you can enroll anytime during the year to our program, and we have lots of different training activities to help get you started. We have virtual training courses through Moodle and lots of other resources to kind of help you go through the step-by-step. And of course, you're always welcome to email anybody from the PAMF team, like I said, at pamf@glc.org. We'll help you get started. But just a shout out, if you are interested or know anybody who's interested in joining the PAMF program, we actually have a live virtual training this Thursday from 10 to 12 Eastern Standard Time. You can register on Eventbrite. If you go to our website, there'll be like an register for participant training. So if you just search like Great Lakes Phragmites PAMF training, this will come up. Or if you want to send a message to me, I'll be happy to, send you the direct link to registering for this event. But besides PAMF, if you're also interested in just learning more about Phragmites management and research, you of course can check out the other resources we have on our website, which include a lot of management guidance. And of course, we have recordings of all of our past webinars. If you have not already, feel free to join any of our listservs. And these will connect you with hundreds of other professionals who work on Phragmites, and you can sign up for our biweekly newsletter as well. And additionally, we have something we call the quarterly research roundup, and it sends out relevant academic papers published from the previous months. And all of these different newsletters, listservs are on the front page of our website, which is here on the screen. And like I said, additional to today's webinar, it is recorded and will be uploaded to our YouTube channel where you can access it for the future needs. And any of our past past webinars are also saved on our YouTube site and posted to our website. We've given a lot of really great talks in the past. I really encouraged you to kind of dig through and see what other webinars we've done. We have really great presentations on there. And then of course, you can connect with us on Twitter and Instagram. We love people stopping in to say hi and see what work they're doing. So tag us in any of your Phragmites related work. And you can find us at phragmites@glc.org. And then our Instagram or Twitter is @glfrag. Okay, that was the fun stuff. And here's a little bit more of the housekeeping stuff. So in order to minimize any potential interruptions, we ask that all of attendees are going to keep themselves muted and as well as their camera off to save bandwidth. If you have any questions or you're having any technical difficulties with Zoom, you're more than welcome to send a chat, and you can send the chat directly to, I think it just says the host, which would be me. And you can let me know what tech problems and I'll try to troubleshoot that for you. And throughout today's presentation, we encourage you to type any questions you're going to have for Nick into the chat box. You don't have to wait to the end. You can do it as they pop into your head. There's a little chat function here. Type it in. We'll have a specific Q&A at the end and we'll read out your questions. If for some reason you have to leave the webinar early and you are sad because you didn't see your question answered, like I said, it is recorded, so you're welcome to check out the recording and, hear your question being answered. All right, that was my little spiel, but why we're here today is because it's my pleasure to introduce today's speaker. Nick is a professor of biology at the University of Wisconsin-Whitewater. His research interests include aquatic plant systematics and invasive species, and recent projects include molecular studies on Japanese knotweed, water hyacinth, and hydrilla. And we are very happy and happy to have him and sharing his work with us today. So I'm going to let you take control and start sharing your work.
Great. Thank you, Teresa. Thanks everyone for joining. I think I need the screen back. Do I? All right, I'm just going to charge ahead. Perfect. All right. Give me a big flag if this is not coming up.
Yep, that looks great.
Wonderful. Okay, well, thanks everyone for joining. I'm pleased to talk about the work that we've done on Phragmites. My journey on Phragmites has been over the last 5 years or so. I've had a career of basically turning molecular tools to increasingly more practical questions. And I really enjoy studying invasive species, things like that, that are open questions that have a nice, clean answer we can get using molecular tools. When I say molecular, usually talking about something like DNA, so DNA sequencing, or I'll talk about the related technologies. This particular project is in collaboration with some colleagues that you can see there. Maureen, I've worked with at Wisconsin DNR for a few years, and she is a nice contact, very helpful in getting me funding and samples from across the state. And then more recently, I've hooked up with, gotten into contact with Dan and Julia from University of Minnesota. So we have kind of a 2-state collaboration going now. I suppose we could add a state if you've got one you'd like to join on with. And then, like I said, using molecular tools to try to get at the questions. Leading question was sort of, what's the genetic diversity? That's an open question for invasive species. And we generally think of genetic diversity as providing the raw materials for adaptation. So adaptation to different habitats, adaptation to evade herbivores or herbicides, excuse me, something like that. And so knowing about genetic diversity might give us a more targeted control approach. And in the context of Phragmites, as we'll see, we have these two subspecies to worry about, and telling them apart could be a challenge. So for this audience, I don't expect I have to say a lot about Phragmites. It's one species, Phragmites australis, and in our area, it's two subspecies. And the troublesome subspecies, this non-native subspecies australis from Eurasia, is one that's really unmistakable. So if I encounter people who aren't even botanists or not generally thinking about plants, I can describe this to them and feel pretty comfortable that they know what I'm talking about. Like, have you ever seen that 10-foot tall grass on the side of the highway? And sometimes they know what I'm talking about. Anyway, this is an example of how it can grow. It grows very densely. It's enormous for a grass, enormous for a herbaceous plant, and creates some problems with competition for natives, creates some problems for animal habitat, and so on. And one that's definitely worth studying. These are some maps from the Plants of the World online. And I enjoy those maps because they give you their political boundaries, but they give you a sense of native and non-native ranges. So the two subspecies, as I mentioned, the one that's native to North America, northern North America, we'll stick with that one for this talk. It's called subspecies americanus, fittingly enough. And it's native to most of the United States and Canada. That's the green native range there. And then the non-native is Phragmites australis, subspecies australis, which is native to Eurasia and other parts of the Eastern Hemisphere, and has become invasive in North America mostly, in all those purple zones that you can see there. And for convenience, I'm going to use these color codings when I've got images or other descriptions of the two subspecies. So just a big orange circle for subspecies americanus and a navy blue circle for subspecies australis. And as you can see, of course, those are present in the Great Lakes. If we take more of a point approach, this one is from iNaturalist. As you get data for these subspecies, you don't always have confidence that you're getting a designation down to the level of subspecies. So in fact, for many years, up until just a little bit before the year 2000, there were, we just knew that we had this species, Phragmites australis, and it was only around that time that the molecular tools allowed us to really discern that there were different subspecies. One was native, one was non-native. So it's important when you're looking for data to make sure that you have the distinction between native and non-native, at least if you're going to draw a map like this. So what you can see on here are the ranges of the native and the non-native are pretty similar. Again, very abundant in the Northeast, the Great Lakes region. And the abundance doesn't quite come through on here. These are reported data from iNaturalist. Usually people take photographs of plants they encounter, and this allows them to designate which subspecies. And so I picked specifically subspecies americanus and subspecies australis, not just at the species level, which wouldn't tell them apart. We can see that there are more points for subspecies australis. This map, I think, maybe helps it out a little bit more. So I made that point about data not always differentiating between subspecies. And so we can't trust every one of these blue dots on here is actually subspecies australis, but if it was, I wouldn't be too surprised. Subspecies australis has really been just taking over. It grows in very similar ecology to where subspecies americanus does. And it's just a more aggressive plant. It gets around faster, it's a good competitor. And so it is really running the risk of displacing our native Phragmites, in addition to all those other ecological issues of competition and wildlife habitat. We run the risk of getting rid of our native subspecies of this plant. And so, of course, I'm based in southern Wisconsin, but this also happens to be a really good place to study these interactions between subspecies of Phragmites, because we're at a place where the non-native has moved in not as long ago. It hasn't been dense here for as long. And we still have a decent number of pockets of the native subspecies. So it's good for study, and it's also kind of increases the importance of studying these plants and maybe trying to declare some kind of line where we can hold one off and preserve the other. Okay, so again, this audience, I probably don't have to tell you much about how to tell these subspecies apart, but I'm going to go over these anyway, just in case, and I guess for posterity, if we got this on video. So these are some slides showing the general morphological differences for telling the native Americanus apart from the non-native Australis. This one I think is probably what I normally would notice from the farthest distance. You can tell it this is a similar time of year to right now. You can see plants over winter, and even in their dried state, have pretty big differences generally. So generally very dense, tall and thick inflorescences in the non-native, and more sparse, not as massive plants, and then thinner inflorescences in the native plant. And so you can also kind of see the ecological harmony, I'd say, is a little bit greater for subspecies Americanus. This is one that has evolved to grow with our native communities, and it's not a terrible competitor, not at risk of displacing other species quite so much. As I mentioned, the flower heads, this is pretty easy to tell from a long distance. The subspecies Americanus, pretty loose, few flowered, and then Australis, dense and many flowered flower heads in the flowering season all the way through the overwintering. Other features, the ligule, this little flap of tissue at the point where the leaf blade meets the sheath. It's a little bit longer in Americanus than Australis. There's some coloration differences, so there's a little bit more redness in general to subspecies Americanus. And then the sheaths, so the leaf of the grasses also consists of the sheath that wraps around the stem. And in subspecies Americanus, that sheath tends to detach during the overwintering portion or in the late growing season, whereas in subspecies Australis, the sheath stays intact and doesn't have that redness to it. So with some practice, you can identify your two subspecies pretty easily from each other. This one probably less likely for you to notice, but if you did get the florets together, this is a grass spikelet, so a small collection of flowers, these bracts called glooms at the base, and they have differences in their lengths that can help you tell the subspecies apart. Generally, leaf coloration, a little more blue-green in Australis versus yellow-green in Americanus. And I mentioned these plants have different ecologies, right? So we have plants that can take over and grow in the same location. And I like this study. This is a nice illustration of the plants growing together and how one subspecies can do better than the other in competition. So just to orient you to this graph here, this is a competitor, a focal competitor in bold there, which is where the measurement came from. And then the plant that it's competing with is in the normal typeface there. So invasives growing with invasives still grow really well. Invasives growing with natives grow pretty well. So we know that the invasives, the non-native plants, grow very well, and even with competition. So even if you grow them densely with other Phragmites australis, subspecies australis, they still do okay there. And then moving down from this, the next two lines are the native. So the native growing with other natives still grows less well than the non-native subspecies. And then the native growing with the non-native grows the worst of all. So we can see the difference between the third and the fourth lines here shows us that the native plants are taking a hit by being in competition with the invasive plants, the non-natives. And if you follow this through over many years, this kind of competition can displace the native that is less of a competitor than the non-native. So I'd say land managers probably would not be surprised to know this, and I think we all kind of know how this probably shakes out. It's pretty obvious which one is a larger, denser, more aggressive plant. So we know we have the two subspecies. And one of our open questions, in addition to just kind of getting a handle on their genetic diversity, the open question is, do they hybridize? And hybridization in invasive plants has been a question of concern for a lot of different invasive plant groups. So for example, here, this is the hybrid cattail, which is known to grow in dense monoculture, practically a single species over long stretches of wetland habitat, to the competition of anything else that would have grown there, again, altering wildlife habitat and so on. So this is an example where the hybrid is an aggressive plant, much more widespread, as I'll show you. We don't have a lot of data to show Phragmites hybrids yet, but if you have two subspecies that are compatible, that can produce offspring together, then the stage is kind of set for hybridization. And so this is important to kind of stay vigilant and keep screening for hybrids in Phragmites. So hybrid cattails, and then hybrid water milfoil was another example that came to mind. This is a pretty good analog for the Phragmites situation. We have a native species, the northern water milfoil, a non-native species, the Eurasian water milfoil. The invasive Eurasian water milfoil is already problematic. And then, as this article title shows you, the hybrid between the native and the invasive is actually worse of basically a worse invader than just the non-native plant. So hybridization, I don't really have any particular slides on this, but just the concept of hybridization allows for a new genetic variation, new genetic combinations. It sort of increases the toolkit that plants can adapt with, and gives them a potential to adapt into growing more aggressively, escaping management, figuring out new ways to exploit dispersal and so on. So knowing that this is possible, it's important to kind of be aware of them and also to try and prevent hybridization. So Phragmites, two subspecies, definitely lots of opportunity for hybridization. So this is an image by Kristen Saltonstall, who's really led the charge on a lot of the molecular evidence for identifying Phragmites, for deciding where the boundaries are on species. And a nice illustration here of the two subspecies we've been talking about growing next to each other. So if you ask the possibility, is it possible for them to hybridize? Well, they certainly grow near each other. These are wind-pollinated plants, as many grasses are. And so the possibility that pollen from one species ends up on the stigma of a different subspecies, should be lots of opportunity for that. These plants are pollinating with clouds of pollen, basically. And it's in some ways, it's kind of surprising that we haven't seen as many hybrids as we might have seen between these two subspecies. All right, so I don't know if I have too much more to add here. So they are genetically similar, and before the evidence really came out of natural hybrids, there was evidence that they could be crossed artificially. So it would be, in some cases, subspecies are not genetically compatible to make offspring. And this just established that they can, in fact, make viable offspring. And as I'll show you also, we have evidence that they're producing viable hybrid offspring in natural settings. So these are the examples to date of confirmed hybrids, what I would consider confirmed hybrids between the two subspecies. And the background dots are just the distribution of Phragmites australis, not really designated to subspecies, but gives you a nice idea of the density. So again, in the Northeast, the Great Lakes region, this is really the home for Phragmites, whether that's native or non-native subspecies. And these plants are a little more sparsely distributed if you go out west. And so we have just these examples of hybridization. And another point here is that I think there's been a lot of surveillance. So I think a lot of people would be excited to report about hybridization. There just hasn't been a lot of it that's really risen to the top in the investigations. So why haven't we seen more hybrids? We'll leave this at the realm of hypotheses, ideas why there might be less hybridization. Here's some ideas that have been forwarded. So phenological separation would be the idea that they are compatible, but maybe not always pollinating at the same time, for example. So if you have one subspecies with pollen available early summer and another one later in the summer, then their pollen from one subspecies would have less chance, even if they're growing in proximity, of pollinating the other. Limits of molecular methods. There's a possibility that you could do a screen using molecular methods and still not detect hybrids. One point to that is a lot of the early data for identifying Phragmites came from just a DNA identification from the chloroplast. And I've got a graphic I'll show you in a little bit here that when hybrids are formed, we typically think that the chloroplasts are all getting inherited from the maternal, the ovule parent. And so if you have a hybrid, it doesn't get two kinds of chloroplasts, it gets the chloroplast that comes from the ovule parent. And so if people were screening for identifying their Phragmites based on chloroplast DNA, that would completely remove the possibility to identify hybrids. And so that's one of these limits. Another limit would be potentially the differences that become clouded by repeated crossings or something like that. And then this third point is just a kind of a plug also for our region. So the Great Lakes region is a little bit newer to the game, I think, of studying Phragmites. A lot of the early efforts were on the East Coast. And so, like I said, this is an important place to study Phragmites. It's also kind of a current investigation to learn more about Phragmites here. Okay, so I showed you the points on the map where hybrids have been confirmed. One really open question, and maybe one of the bigger questions that people run by me, is sort of, how do I know if I've got a hybrid? And I would say that the data are sort of inconclusive at that point. There's not a single identifying hybrid morphology to watch out for based on the evidence so far. But with hybrids, you typically expect somewhere in between the parents is often your expectation. So if you have a measurement, big and small, the hybrid might be somewhere in between those. However, that's not a rule either. So you hybridize two species or subspecies, you can have hybrids that have features very much like one parent or the other parent. So this is one hybrid that was identified in New York State and highlighted here in the bullet points there, the features that are present on that hybrid and how they track to one or the other of the parents. So it has a couple features that would be mostly like the native Americanus, features that are mostly like Australis, including infection by this non-native gall midge. And then a feature that they declared was basically intermediate. So all possible combinations. And I guess if anything else, this would be, if you were careful about observing your plants in the field, this could be something that tips you off that you have a hybrid, finding an intermediate measurement or a combination of traits, some traits tracking to one parent and some tracking to the other parent. This might be a good reason to study that kind of plant in more detail. Okay, so this is from the New York study. The other study that had good data about the morphology of the hybrids was from this cluster of hybrids in Nevada. So quite a few hybrid plants were recovered from the Las Vegas area. And they had two kinds of hybrids in their study, some that had a maternal Australis parent. So the non-native plant is the ovule parent. So it gives the chloroplast in the arrangement. And plants of that type, according to their data, were very much like the non-native. So they were resembling their maternal parent. And they also had plants that were tracking to the native plant as its maternal parent. So the other kind of hybrid, or reciprocal hybrid. And that had a different morphology. So one with the Americanus maternal parent had this kind of unique, separate morphology of being small and skinny. So altogether, just all of these examples, we have very few hybrids across the country. We have, even in the studies where hybrids are found, they're a one-off or just a one or two per study most of the time. I don't know if we've really built a big base of information for knowing what's possible with these hybrids, but certainly a lot of variation and a lot of uncertainty about what the hybrids kind of should look like. Okay, so I'll go through this probably pretty quick, but if you needed some information about the DNA of hybridization, here's my little image of plant cells. And so if you're hybridizing, this is a mating event between two different, usually species or subspecies. And mating involves half of the DNA from the nucleus from each parent. And then, as I mentioned before, the chloroplasts, we'll just keep it nice and simple here. We'll just do the majority outcome is that usually most of the chloroplasts end up in the offspring. So if we have a cross between an ovule parent and a pollen parent, the ovule parent donates all of the chloroplasts and their corresponding DNA, and half of their nuclear chromosomes, and the other half of the nuclear chromosomes come from the pollen parent. I have a little disclaimer down there. They're a whole other concern if you worry about the chromosome number in species. We're just going to skip over that. So playing with 6 chromosomes here. So just to this point, if we can identify the DNA, then if we have a hybrid, we should be able to see in the nucleus, if we get some kind of nuclear DNA, we should see half of that coming from the one parent species or subspecies, and another half coming from the other parent subspecies. So I showed you the map where these come from. This is an image showing basically the evidence for what they used to decide or declare that they had hybrids, in fact. And most of these are based on microsatellite data. So I'll explain that on the next slides. Microsatellite data are a way to get alleles, basically. So if you go back to your genetics class, for one gene, basically one comparable piece of DNA, you get one allele from dad and one allele from mom. And again, if you have a hybrid, you get one allele from one subspecies and one allele from the other subspecies. And in a lot of cases, those subspecies have unique alleles that you can say, this definitely came from Americanus and this definitely came from Australis. The microsatellite evidence is 8 to 10, usually, of those genes, basically. So sets of alleles. So it's nice and robust. It's got more than just one gene worth of evidence. And that's kind of why these data have to be portrayed in this way, is that you can't easily show 8 ways of variation in the same graph. So this is where a lot of these are showing principal components analysis. So these scatter plots are principal components analysis. They're basically a summary of genetic variation, and individuals that are more genetically similar are dots in the same place or close to each other. And you end up with kind of a field of variation. And in this case, this is the native subspecies, this green triangle field of variation, the red circles for the non-native subspecies. And then the evidence for hybridization is these points in between that aren't cleanly assigned to the native or the non-native cloud. In fact, there are a lot of them, you could think of them as kind of right in the middle there. So in between on this first principal component, PCOA1, similar thing here. Sorry, I can't see everything because my people are covering the, hold on, let's get that. There we go. All right, there's our hybrids. So suspected hybrids here now, circles in between triangles of invasive and pluses of native. So very similar here, several points for the Lake Erie study, 1 point here for the New York study. And then this one is a structure analysis. So if you're not familiar with that, a structure analysis is a way to take, again, abundant data and to assign, sort of statistically assign individuals to populations. And the heights of these lines, it's a bunch of tiny lines all stacked together. And the color of the line is the confidence that you are assigned to one subspecies or the other. So green subspecies, most of these are 100% assigned to green subspecies. I'm sorry, I forget which one is which. In the red zone, we have 100% assigned to red subspecies. And then this arrow is highlighting an individual that is just about 50% uncertainty. So uncertainty, 50% assigned to green subspecies, 50% assigned to red subspecies. That kind of evidence is enough really to run with and say, this has got to be a hybrid. It's got half of its DNA looks just like Americanus and half of its DNA looks just like Australis. So mostly evidence using these principal components plots or structure plots. I'm going to show you the same kinds of data using our data as we get farther along here. Also want to put a little plug in here. Wendell and colleagues have a method that also uses nuclear DNA. And this is based on a single gene. So sometimes you can get a clean answer with just one gene, and you would expect that same pattern. So if you have a first generation hybrid between one subspecies and the other subspecies, you should have the DNA form of the native subspecies, which is these two lines that get cut by the restriction digest. That's present in the hybrid, as well as this other line for the DNA that does not get cut in the introduced. So the hybrid here is showing us a combination of the introduced DNA data as well as the native DNA data using just one gene. This one has an advantage of being probably faster, simpler for a single plant if you want to check it out, or a handful of plants. These other big data studies are a little bit more labor intensive, a little more costly, I would say. Okay, so I gave you a lot of evidence from microsatellites. These were the first evidence in Phragmites, really the first data we had for genetic variation. So again, taking all these effectively like a gene. One of these markers is kind of like a gene, and it has alleles, which are differences in length. So a microsatellite is a piece of DNA somewhere in the genome that has a run of nucleotide letters that repeat. And by the process of copying DNA, natural process in a cell, if you've got a set of repeats, the cell tends to sometimes copy too much or maybe copy too little. And so this length can vary over smaller amounts of time than like big evolutionary time between species. So within one population of the same subspecies, you can get some variation where they have different lengths in size. So you have the standard PCR primers, which are these purple zones, kind of tell you where to amplify, and then the size of the piece of DNA that gets copied, this PCR product, that size depends on how many repeats are in that green region there. So hopefully that clears up microsatellites a little bit. There are 10 microsatellite markers for Phragmites. We ended up using 8 that I find to be more reliable. And then even within that, this PAGT12 is sometimes a little troublesome. This is a summary of the data from Saltonstall, year 2003. Basically, the first big evidence of genetic diversity in Phragmites, and she found these alleles are a lot of them private to one subspecies or another. So I mentioned my coloring scheme. This is kind of a variation on the coloring scheme where orange is still kind of my preferred color for the most common alleles in the native. So subspecies Americanus, orange has an allele and has this number. So this is a length size for that allele. If it's unique to Americanus, it gets orange or red or yellow. If it happens to be shared, so these can be shared, for example, if they both inherited it from the common ancestor and it's just not variable between subspecies, sometimes they get the same color. So this 178 is found in Americanus. It's also found in Australis, although as you can see, very different frequencies. And then down on the bottom, there are alleles that are unique to Australis, mostly in this blue color, sometimes green. So same thing there. So if you get one of these that's unique to Americanus, even just one of these alleles in your sample should tell you that you've got DNA from Americanus. And so sometimes they can also be diagnostic to subspecies. And then using 8 markers, you get even more confidence in the identification. Here's the same graph as before with the inheritance of those alleles. So just imagine that your data are somewhere on these chromosomes, and this is a cross between 2 members of the non-native subspecies Australis. One's got these 2 alleles, so just like genetics class, 2 alleles. This is homozygous 198, 198. This one's heterozygous 198, 202. And in inheriting those, this one can pass on one or the other of its alleles, and this one passes on one or the other of its alleles. And we still got alleles in the blue or green Australis color palette there. So this is a cross within a subspecies, a natural cross. If you have an instance of hybridization, then you would take DNA from one type of parent and mix it with DNA from another type of parent, and you get, in this case, potentially an allele that's distinctive to Australis combined in the same individual with an allele that's distinctive to Americanus. And so this would be evidence of hybridization in that individual. Again, you can get evidence using just 1 marker, and then if you get that same signal from 8 markers in the same individual, you have high confidence in your declaring that to be a hybrid. Okay, so we did microsatellite analysis on Wisconsin plants. These are some summary images of the data we got. This was published, I forget, a few years ago. And the image is showing an assignment to sort of hypothetical populations. Tried to use the same palette, so oranges and reds for the native and blues for the non-native. And it's kind of all together, if you sort of lying on top of each other, that we had both subspecies kind of mixed together in this image. But it does show you that we've got more predominance of the non-native closer to Lake Michigan. That's consistent with what we see in the field, of course. And then these more isolated populations tend to be more of a native subspecies that hasn't been pushed out yet. Comparing to those other types of data we saw before in a principal components plot, we've got a pretty clean Americanus cloud here and an Australis cloud here, and no points really in between them. So we concluded in that study that there's no evidence for hybridization, whether you look at the principal components plot or the structure type of data. There's some uncertainty here. There's a little bit of missing data that I think is accounting for this, but no individuals that are like a 50-50 assignment to both subspecies. So no evidence for hybridization, even though we looked in a lot of places with a lot of individuals per population across the state. And then, as I said, we added on the, so I had done the Wisconsin data, and then we were able to add on the data from Minnesota, where they also did a study similar with microsatellite data. I'm going to go a little bit faster through this. This just shows that our data from Wisconsin and Minnesota was very similar to the North America data overall. Like I said, I don't know if I trust this PAGT12, so we'll kind of ignore that one, but everybody else is lining up pretty well with what we expected for the native in orange and the non-native in blue is pretty similar, even just the abundance of those alleles. So this tells us that I guess we're taking a pretty representative subset of even North American plants to get the data. Principal components with the combined Minnesota-Wisconsin data, starting to get some points that creep into the middle here. The way the data are portrayed here, there's a label, which is the centroid for points from the same population. So all these points are from the same population. They're kind of clustered together. This is a population from Minnesota that's kind of creeping over from the Australis zone into maybe a little ambiguous territory between subspecies. And then a few points from Americanus are creeping into that middle zone a little bit. Lots of variation. We found, interestingly, really higher genetic variation in the non-native subspecies than in the native subspecies, which not what I expected going in, but that's what we got. So we also had lots more points for the non-native, but some of these big circles here, this tells us that we had basically genetically uniform, as far as we could tell, individuals of subspecies Americanus that are all kind of stuck together in that small genetic variation. Looking at the data on a structure type of plot, this is the microsatellite data, and there's a zone here where the assignment to populations starts getting a little more like what we'd expect to see in a hybrid. So around here, we have individuals that are 50% blue assignment to native and orange assignment to non-native. This tells us that maybe we've got some hybrids going on there. With the microsatellite data, as I said before, there was some missing data, and I think that might be one reason that we might not 100% trust that. So we kind of had this suspicion going in, and went forward and got some higher density data for Phragmites in Wisconsin, Minnesota. I mentioned that the microsatellite data we have effectively like 8 genes that can be compared, and 2 alleles per individual across all of those. Time marches on, technology improves, and the newer technologies are genomic level, and you can do this data gathering called genotyping-by-sequencing, where you get a lot of genes. You don't necessarily know a lot about them individually, but it's enriching for places in the genome that are variable. And so basically you end up with those as alleles, and we go from 8 alleles to about 7,000 alleles. So the power to detect variation, to assign individuals to their closest relatives, the power has gone way up with this new technology. The broader category, we call it genotyping-by-sequencing. And then the company we've used to get the data is called Diversity Arrays Technology. They're in Australia, and they named their data, they call it DART-seq. So I'm going to use that acronym there, DART-seq, to talk about the data, and just a little graphic of what we can expect to see from DART-seq data. Again, it highlights places where there are differences. So it might be the case where you have a difference that's present in Americanus and absent in Australis. And so one kind of DNA here and one kind of DNA there. That's one piece of data to tell you about how different they are. And this kind of evidence is good for telling them apart. Kind of build your confidence that a site like this would have orange always found in Americanus and blue always found in Australis. And then with so much data, there's also the possibility that you get pieces of your data that aren't maybe not as informative. So here's a case where this allele might be present only in Americanus, but this individual is heterozygous, has one but not the other. Sometimes there's just a pattern that doesn't line up, but in the area of big data here with this big principal components plot, we're going to take all this variation and kind of look for the biggest picture we can find. So there might be these other 2 kinds, but overall, we're kind of expecting to see a signal that tells us most clearly that we have one subspecies or the other. All right, hopefully you're still following me on that. So lots of sample locations, pretty good overlay of the places we got samples for the microsatellite data and places where we got samples for the DART-seq data. This was the identification going in, so a good spread of natives and non-natives. Big cluster around the Twin Cities area where the non-native is pretty commonly found as well. The result from the DART-seq data is we got, I'd say, a much more substantial cloud of these intermediates in between the orange cloud and the blue cloud. And I'm using those same shapes again, where squares are Minnesota. So the Minnesota data are filling in this in-between space, quite a few. So plants that we basically visually identified as Americanus are filling into this in-between space. Some of these individuals that we visually identified as Australis here and here, also filling in that middle space. And importantly, my colleagues in University of Minnesota we're able to get some DNA from these known hybrids in other parts of the country. So these plants from Las Vegas area that are known to be hybrids allows us to get a lot more confidence that this method is detecting and showing us what we expected to see, actual hybrids. So the known hybrids are in between orange and blue. Now we've got suspected hybrids that are a lot of points from Minnesota now. Roughly comparable, I'd say it's not quite as clean in the microsatellite data to get these points that we're feeling are hybrids using the microsatellite data. The data are not one-to-one, so there's no guarantee that every point in here is represented by the microsatellite data. But a little bit better, I'd say much better confidence with the DART-seq data on those being hybrids. If you compare sort of total up all the sample sites for microsatellite data and DART-seq data, we get a similar pattern to what we saw before. So now this block, potential hybrids doing the same thing where they're assigned 50% to one subspecies or the other, or somewhere kind of intermediate. So with hybrids, if there was any kind of back crossing or introgression, you could see different proportions of their genome being belonging to each parent species. But at least some individuals, pretty good chance of being hybrids. And then this is what I put this together to illustrate what I was talking about, that a lot of the individuals that we have DART-seq data for that we suspect are hybrids, those aren't actually represented in the microsatellite data. So pretty strong evidence for hybridization. And then here, it's still a little uncertain on that one. And then the known hybrids from Las Vegas, these come up with pattern very much like we would expect to see. So just about 50% assignment to both subspecies there. So as I suggested, I'd say probably more power to determine that we have hybrids using this newer and fancier technology, DART-seq. And we're actually in the middle of getting new data for DART-seq that didn't quite get back in time for this talk. So our suspected hybrids, not really a geographic pattern, pretty widespread, or peppered in among all the other populations that were sampled. This is where they were found. Going back to revisit those populations, as I mentioned when we talked about the hybrid morphology, sometimes it's clear, sometimes it's clearer than other times. These are cases where we were kind of surprised or maybe just sort of generally observing lots of different ways that the plants can appear if we know that they're hybrids now. So, or I should say suspect that they're hybrids. And they got morphologies all the way from looking very much like Australis, as this dense patch looks, very green and not red. And then this is another patch where the morphology is looking a lot more like Americanus, also came out as a hybrid in our study. So kind of consistent with the evidence that we don't really know what to expect from the hybrids. Okay, so moving forward, there are also reasons to be cautious about our DART-seq data. I presented on our results earlier a few months ago, and I'm not ready to make an absolute declaration that we have so many hybrids, and I'm excited to give it a chance to corroborate the data. So we got some new funding for this year, and we pushed through some new sampling using DART-seq, and we haven't got the data back yet, but we should be able to take these yellow sites that were determined to be hybrids. We have new plant collections from those sites. We're running the DART-seq analysis newly on them. So if we continue to get evidence that that's hybrid at these yellow sites, then I'll feel more strongly about that. And yeah, we'll kind of see how it goes. So if you want to know how that turns out, I don't think we're too far away. You know, we're talking like weeks until I get the data. I just didn't have it in time for today. So if you're curious about how that turns out, let me know. I'll keep you in mind. Can let you know how it goes. But at this point, I'm really waiting on that to know, to feel better about saying that we definitely got hybrids, especially on this scale in Minnesota. But certainly some suggestive data at this point. Okay, so I'm running close to the end of time there. Let me wrap it up here. So if you're looking to do any kind of microsatellite or DART-seq analysis, I would say the cost kind of comes out the same. The DART-seq, you have to run 94 samples at a time, and that's in the thousands of dollars. But the per sample is not too bad. And the cost there is increased for the analysis for DART-seq. The cost for microsatellites still pretty labor intensive in the laboratory. So I just really broad estimates, but I would say that the cost per sample, if you run them at scale, it's kind of comparable, microsatellite and DART-seq. And the DART-seq data, I'd say, is more pleasing to run analyses on, and it really gets rid of the ambiguity. We sort of outsource the data analysis to somebody else. So we have the ability to do more. This is a little plug for myself as I'm winding down here the talk. This past year, I did a good amount of basically custom genetic testing. So if people have plants, really plants of any kind, I have a service, I've named it Phytoid or Phyto ID. Kind of still taking off at this point, but for a fee right now, we're working at $45 a sample. You can send plant material that is difficult to identify, or you just want a confirmation, and I'm offering to do that in my lab. Feel free to get in touch with me if you want to do that. This is a good resource for people who are managing, for example, aquatic plants, or any case where you might have suspected hybrids. We can throw some of this DNA technology at it. If you wanted to do something with Phragmites, a DART-seq analysis on Phragmites, then we'd probably be looking at something at a much bigger scale. Doing 1 or 2 samples, I don't know if that's feasible, but it would be possible to run a large number of samples in some kind of a, I don't know, funded operation. All right, so keep me in mind if you have any unknown plants you want DNA tested. And that brings me to the end. As I mentioned, I've been on this project for a while, had a lot of help from people, and I really appreciate all the help from collaborators who are getting me samples, helping me with the analyses, certainly helping with the funding. Yeah, I have a lot of undergraduate students who have been working with me at UW-Whitewater, and excited to see where this goes in the future. So thanks for tuning in.
Thank you so much.
Thank you.
I'm going to share our screen. And I just want to encourage anybody to use the chat function, the little, looks like a little messaging box. Put them in the chat. We'll do I know it's kind of close to the 3:30. So if you need to leave right at 3:30, put them in, we'll answer them. And then hopefully you can check back the recording. But it looks like we already have some questions. So if you don't mind, we'll just start going for them. Oh, excuse me. You guys don't want to see that. Okay. It looks like we have a question from Doug Wendell. What is the form of data you get back from DART-seq? Do you get all the I'm assuming this means something smarter, SNPS?
Yep. All right. Yeah, I almost went through that term. SNP is single nucleotide polymorphism.
Okay.
It's a whole lot of syllables to say, if you have the DNA letters, A, C, G, T, in the sequence you compare, there's mostly, if you compared like 2 people to each other, we'd mostly have the same DNA sequence. And the SNPs are locations where sometimes one of your letters is different. And so that's the single part. And so if you had the example I showed with the 2 orange and the 2 blue, that would be a single nucleotide difference where the orange both have A, for example, and the blue both have C. And that is, to answer the question, thanks, Doug, that's the kind of data we get. There's a sequence where they give you the flanking region, so that gives you kind of a unique identifier for that piece of DNA in the genome. So these big genomic investigations, they just like take a bunch of DNA and they throw it at a computer, and the computer says, oh, I found these pieces that match, and they match everywhere except for this one place. And so that's the SNP, the SNP. And so we have pretty good confidence that it's a comparable piece of DNA because everything else matches. And so sometimes you get all A's in one subspecies and all C's in the other subspecies. And then sometimes you get some variation. So it's a good, it's a whole bunch of places where there's variation, sometimes between subspecies, sometimes within a subspecies. And it's, I guess, a little tricky that it's also sort of defined, I think they define it to sort of 2 nucleotides at a time. So you get this either a binary presence absence of the A nucleotide, for example, or you can get a 0, 1, 2 for a homozygous, heterozygous output. But it's in the like the bioinformatics realm of things. So they give me a giant spreadsheet. They've also written a computer script that can help to kind of parse that out. And yeah, it's sort of in the realm of, we let the computers handle it because it's really hard to visualize those, even a few at a time. So you just throw them all at the wall, and that's where you get your PCA plot.
Awesome. And Doug, if you have any additional questions to his answer, you're welcome to unmute. But I'm going to go to the next question from Keith. Is there something about Las Vegas environment that might be, oh, he does have his hand up. Go. Yes, Doug.
If I can. Oh, hi. Yeah, I'm sorry, Nick, thank you. It was good talk. I remember at a conference, I think it was last fall, you said that this company's sequence was, there were some proprietary aspects to it. So I was just wondering if you had free use to do all, can you do all you want to with the data you get, or are there any restrictions on what you can do? Because that's kind of what I was driving at.
That's a good question.
Okay. I can do all I want to do, which is to have some confidence about, so I gave that number of 7,000. I feel pretty comfortable that those are 7,000 unique places in the genome. If you wanted to replicate anything like that, I don't know if the sequence, if you wanted to like get a DNA sequence on your own using like Sanger sequencing, I don't know if you could do that easily because it's a fairly small flanking region. But with more genomic tools becoming available, if you had like an annotated Phragmites genome, you could take all those points, or at least a lot of them, and assign them to chromosomes and assign them to genes. I'll be honest, I'm not really immersed in bioinformatics. I feel like I'm just a little bit behind generationally. Younger people would love to do that for us. But yeah, I'd say that there's a lot you can still ask, and I don't think the data would be so much different from what you get on your own. Their technology, they've brought the costs down by like 4 times over what somebody else would offer to do it. So that's why we're working with them. Their data, it's very affordable for what it is.
Great. I think that was a helpful clarification.
Cool.
Another question, is there something about the Las Vegas environment that might be more conducive to hybridization that we're not seeing in other regions?
Right.
This might be out of the scope of your research.
I don't know if I can answer that. I would point you to the study. The question about them being a clone, their data points are not 100% overlapping or identical. So, if you trust your data generation and you have a clone, I would say your clone should be 100% identical across microsatellite markers if they were in fact clones. And I believe that that's probably an epicenter for study because it's a pretty, it's not just one-off. I feel like a lot of the other publications that had hybrids were sort of surprised. We were surveying a whole bunch of plants and we found one that was a hybrid. And this is one from the Las Vegas area where they can say, like, they can take you on a tour and say, look at that whole crop of plants is a hybrid or is made up of hybrids. So I'd leave the possibility that there could be some variation in maybe a lab technique where it didn't come out 100% identical. That could be possible, but the data are suggesting that they're genetically different a little bit, even though they're all hybrids in the same place.
Perfect. A question from Julia regarding the displacement map with the blue and gold icons. Did the data come from midwestherbarium.org?
Oh,
the distribution does not match what I'm familiar with in Minnesota. You can speak up too if you want to add anything to that question, Julia.
Julia's a co-author, so. Let's see. Oh, I'm sorry, I'm on the wrong button here. I was going to try to pull that up again, if that's okay. I think maybe you're talking about this one early on. Is this the one?
Yes, that is the one.
Okay. You might have called me out on this before, and I have it here. I basically took a grab. I thought I had my updated one of EdMat's, so just all the sources I could get, basically. So there's GBIF, the Global Biodiversity Information Facility, that does herbarium specimens. It also pulls from iNaturalist, Consortium of Midwest Herbaria. Sometimes there are points, these are specimens as well. And then EdMaps is more of a land manager's on the ground data source. So I guess if you wanted for our audience to clarify what's not matching with your experience, you're welcome to.
Yeah, we'd have a lot less blue in outstate Minnesota. And the blobber on the Twin Cities, we do have a lot there. In total, we have a little over 1,600 populations that we know are invasive. And we're splitters, not lumpers. So some of those are kind of smaller populations near each other, but we've marked them all for the contractor to find. Who's going to do the treatments. But yeah, we're not quite as rich with invasive Phragmites as that, but.
Right. Yeah. So my little disclaimer there, I don't know that every data source makes the distinction. And I think that's, something maybe we'll catch up on, but if you're going with herbarium specimens anyway, it just would be kind of a blanket Phragmites australis determination. And the way that taxonomy works is that if you don't specify a subspecies, sometimes it might sort of get assumed. So I kind of erred on the side of having more points, even though they weren't all specified, and I didn't distinguish like definitely australis from could be either. So.
It did make me curious to maybe want to go to the herbarium and browse through and maybe make some corrections on their records.
Yeah, that's a lot of data points, but
yeah,
somebody's got the time, maybe.
And we do have a lot of Americanus in Minnesota, given land of 10,000 lakes and all. So there's a lot of that. Obviously, more than shows on that map.
Right.
But the distribution would be appropriate. Like it's throughout the state. You can find pretty much everywhere.
Great. Thank you. Let's stop again.
Yeah, I think I'm gonna, do I have to reshare? There we go. Okay. Okay, a few more questions. These are a few from Ping. I'll just read them first, the first one first. The DART-seq seems to generate thousands of SNPs. If so, are any of the SNPs associated with the functional genes?
So great question. I have not looked at all. I'm always kind of mystified. I think when there's a certain scale, I just like, it blows my mind a little bit. So, you know, you could try looking one at a time. I imagine a lot of those you'd get your result if you tried to like match for similarity to an existing known sequence. You'd get a lot of them returning that there's just no match. Although you can do a computational version of bioinformatics version of that. So I haven't done any of that. That would be interesting for sure, if we could identify traits. I think that would be, if somebody wanted to say you had two subspecies and you wanted to know if there's some kind of adaptive genetic element in you know, like find the origin of invasiveness or something, or the major genetic drivers of that. I think you'd want to sort of focus a small number of plants on getting a lot of data. Maybe someone out there is working on it to get sort of a whole genome level data for Phragmites. So I don't know the answer. I think
work for a future student.
Right. Yeah.
Another question: how do you think the prospects of identifying trait conferring genetic biomarkers from the DATCERC data?
Yeah. So this is kind of what I was getting at. I think you'd want to trait conferring markers. I think you'd want to just start from scratch with a study that, you know, say you're working through genetic variation in Australis and you find individuals that are more aggressive or have some feature that you're interested in. If you had has the traits and doesn't have the trait plants, and then you like threw that at genomic data, and then you find a difference, a genetic difference, that kind of study, like a genome-wide association study, would maybe tell you what's different about them that could be responsible for that adaptive difference. Or even at the subspecies level, you could go functionally and say, I'm going to take a lot of members of Australis and a lot of members of Americanus and see what's different about them genetically, functionally. That's an open question as far as I know. And it's outside of what I would do for myself, that's for sure.
You're getting a lot of hard questions. How do you distinguish between the SNPs and suspected hybrids and natural mutations in each subspecies? I looked at that.
I don't know if there is a distinguishing, except that I think at the genomic level with something like this, you trust in the power of the scale. Mm-hmm. So there are going to be, in your set of variation, there's going to be some sites that are consistently different between subspecies. You're going to have some sites that have randomly mutated to be the same in individuals. But by the frequency of mutations, I think you'd expect, you know, the genome isn't just mutating across the whole genome every generation. So there'll be a good set of the differences that are stable, more stable, and those will reliably tell your individuals apart. And the rest is sort of noise. It's signal to noise ratio. So I think we're pretty comfortable with that signal to noise ratio. That a lot of the markers are maybe not all that informative, but there are enough that are that will come through.
Sure, thank you. And then I guess just one, if you have questions, you can still put them, but we'll kind of end with maybe a higher level question of, have you heard anybody, what are the potential management implications if there are hybrids or potential hybrids for the species popping up in Minnesota and Wisconsin?
Right. Yeah, that's always the next step, right? And I
might not be a clear answer at the moment, but
I find myself on a number of studies that are, you know, looking at the genetics of invasive species or identifying invasive species. You know, we have something similar with Japanese knotweed. You know, identifying them isn't the end of the story, and it's not certain that it's going to point you in the direction of a management control. Yeah. Some thoughts, though, if you found out that hybridization was more of an issue, then you would maybe take a management approach to prevent pollination, cross-pollination. So, for example, like mowing prior to the flowering season, that would remove your possibility of hybridization. The plants are still there, but they're not going to donate pollen to their neighbors. And then another one is just identifying genetic variation of invasives, the potential that we discover some kind of biomarker for being more invasive, being herbicide resistant, being particularly competitive or something. So I think it's an important first step.
Sure.
We sort of peering under the hood of a massive complex of individuals, and we're leaving the possibility open that we'll discover something useful for managing them.
Yeah.
But it's sort of coming back to the questions about biomarkers for adaptive traits. Maybe there's something out there. There's the aquatic plant hydrilla has a marker that shows that it has a herbicide resistance, for example.
Yeah.
So it's just a level of new data. I think land managers, you might end up feeling like you don't have new options, and it's a long shot, but maybe there's some of these genetic options that will turn up a new strategy.
Yeah. The more information, the better, for sure.
Right.
All right. Well, I don't see any more questions, and thank you for hanging on past the hour mark to answer.
Yeah, my pleasure.
And for anyone who, I know we were kind of encroaching on the field season, so if you if you want to share this with colleagues who weren't able to attend today, this is recorded and we'll post it onto our YouTube page, and we'll send it out to the Eventbrite as well that you registered on. So the recording will be available soon. And just thank you everyone for watching. And we want to credit US Geological Survey for our grant cooperative and GLRI for our funding to be able to do this. Thank you so much. I really appreciate, Nick, that you taking the time to present a webinar with us.
I'm glad to do it. Maybe we'll see you again next year.
Yep. Take care, everybody.